Arowor Corp is a biopharmaceutical development company with a drug-centric focus on protein, peptide, vector, and insert therapeutics. We specialize in the broad disease areas of rare and ultra-rare infant and aging disorders and viral and nuclear disaster response. As an applied sciences startup, we work at the trinary manifolds of biology, chemistry, and physics, and the basic fundamental and applied natural sciences. Our effort is to develop and commercialize discoveries and inventions toward the manufacture of regulated goods. We keep scientific pillars such as open and honest discourse, unrestricted data, senior independence and autonomy, and an emphasis on reproducibility, rigor, and stringency. About me, my name is Aron Workman. I'm chairman and CEO. I founded a research development engine at Axoxy Laboratories in 2010 with my first issued patent, and I founded Arowor Corp 10 days ago. I'm going to school for my MS in pharmacy at the University of Florida, and I'll be done in 2 semesters, summer this year. I've published in Journal of Neurochemistry, Molecular and Cellular Biochemistry, and Computational and Theoretical Chemistry. Probably the most interesting thing about me is that my great-grandfather is Thomas Jefferson. The 3rd president is my true cousin. These point trajectories on the map are nuclear hotspots. DCCS developed a computational daemon for modeling and predicting nuclear explosions. That's Project VESUVIUS. Our primary development project at Arowor Corp is RDP-1, a highly complex biopharmaceutical under development for the treatment of acute radiation syndrome following large-scale nuclear catastrophe. One solution to nuclear disaster is an adequate pharmaceutical in the disaster response plan, of which it is currently absent. Amethylstein adjuvant, and there's a couple others with low efficacy and pain points. There's just no outstanding solution for ours or DEAR. There's considerable disaster response planning and finding a way to validate that our drug belongs in stockpile and that it is properly stockpiled. We have a rotating stock strategy whereby the active ingredient in stockpile can be reformulated and turned over for other indications, notwithstanding the near certainty of future nuclear catastrophe. That's all part of the Superoxide Dismutase analog program. Let's take a glance at the industry. Global pharmaceuticals and medicine manufacturing had revenues of more than $1 trillion USD in 2015, with brand names in the US claiming $162 billion USD. The global biotechnology industry had revenues of $312 billion in 2015, with 1/3 coming from the US and 8% from Europe. In 2016, the US biotechnology industry held a market capitalization of $889 billion USD, 131,700 employees, and the leading companies, or those with greater than 500 million in revenue, had net income of $32 billion USD. There are approximately 12,000 biotechnology companies active worldwide, and the global industry differs from the US industry by both relative company count and percent share. About 3,000 companies share 26.8% or $29.8 billion USD of the concentrated US biotech industry, whereas about 12,000 companies share 79.7% or $257 billion USD of the segmented global industry. A major trend in the industry is that leaders control just 1 or 2 revenue-dominating products per cycle. In biotech, a single super high-yield mega blockbuster product is both the majority marketing investment and revenue source for every leading company studied. Increasing population count, median age, life expectancy, and number of physician visits are metrics often cited as long-term driving forces in biotechnology. Another trend is an industry-wide decline in R&D spending. The industry leaders spend 5% to 20% of their revenue on R&D. The largest industry leader reinvestments are acquisitions and marketing. The human health technologies segment is predominantly comprised of therapeutics, and the therapeutics segment is predominantly comprised of therapeutic proteins. In particular, the therapeutic enzymes market type segment had just $6.6 billion revenue in 2015. The lysosomal therapeutic enzyme market is prominently comprised of Cerezyme, Myoxyme, and Fabrazyme from Sanofi and Genzyme. The thrombolytic enzyme market is notably comprised of Activase, Pulmozyme, and Erwinase from Roche. And the pancreatic enzyme market has Creon from AbbVie as a major product. The Arowor Corp pipeline include biopharmaceutical candidates campaigning through the laboratory and preclinic. RSX is a protein therapeutic under development for RS and other unmet disease. RDP-1 is a first-class protein therapeutic under development for those most affected by RS and DEAR given from nuclear fallout. AO10 is a protein therapeutic under development for chronic lung disease in neonates. CHP1 is a vector therapeutic under development for amyotrophic lateral sclerosis. H10N is a peptide therapeutic under development for perioperative usages across surgery. HSP1 is a vector therapeutic under development for neuroinflammation. MIV1 is a vector therapeutic under development for acute flaccid myelitis in infants and neonates. And GTXN is a protein therapeutic under development for as of yet undisclosed cores and electives. H10N is likely our first candidate that will be elected for an IND and subsequent BLA, It's an antifibrinolytic peptide intended for perioperative settings. It won't have the immunogenicity, anaphylaxis, and angioedema associated with apertinin or other existing solutions. It's easily validated with simple process chemistry in animal models, so H10N is our frontrunner. We have regulatory challenges and opportunities, including vouchers and special approval mechanisms. Overall, we are very excited about our field and our candidacies. Here's some of the structure and reactivity of superoxide dismutase, with the pharmacophores I discovered in the middle row and cartoon depictions of superoxide dismutase on the bottom row. Superoxide dismutase is a complex gene product that binds a copper ion, a zinc ion, an interdisulfide bond, a salt bridge, and other molecular specificities. Understanding the structure and reactions in matrix and applying those findings to the laboratory has generated a very fruitful result. It's our trade secret how this protein functions and why it requires a specialized bioanalytical workflow. Drugs based on the superoxide dismutase analog SODA API are facing a third-generation transition. There was previously marketed superoxide dismutase as a pharmaceutical in Europe in the 1980s, not yet in the US, that's a virgin market, except for glycidin nutraceutical and competing products under development. There's some other markets here as well that we're targeting. Like the insulin analogs, superoxide dismutase underwent genetic cloning for a recombinant source so that the human protein is injected rather than the cow or pig or wherever it was harvested. And like the insulin analogs, there are now naturally occurring mutations in SOD1 that increase its bioactivity, stability, and other advantageous biochemistry. These advantageous biochemistries will translate to advantageous clinical effect. Here's what our initial processes look like for molecular cloning at top and process chemistry at bottom. At least 2 of 6 single-variant superoxide dismutase analogs have improved activity, metallation, stability, constitution, and aggregation relative to wild type, which are industry-standard advantageous properties for therapeutic protein compositions. The fastest single-variant superoxide dismutase analog, E40Q from US Patent 373, has activity by the NBT method greater than 2.6 times that of wild type. E133Q has activity just 2 times that of wild type. E40Q is the most efficient superoxide dismutase ever discovered or invented, and is likely even the most efficient protein ever discovered or invented in totality. Furthermore, compound variant superoxide dismutase analogs may have compounded activity up to 12 times or greater that of wild type. The NBT assay has an upper limit of detection of 3 times that of wild type. Before I get into the minutiae of what we need done in the lab over the next couple years, let's look into the future. Series B or C has closed, and we have strong enough data to submit an IND. We have options with licensing or distribution and marketing, but we'll likely partner for those 2 functions, and we'll keep manufacturing and analytical process chemistries on our side. Tasks for the laboratory include cloning, selection, expression, process chemistry, bioanalytics, adjuvant preparations, syringe formulations, pharmaceutics, and more analytical biochemistry. Tasks for the office include research, documentation, data handling, regulatory filings, and other corporate communications. Tasks for the preclinic include up and down studies, no adverse effect levels, and relative dose-response studies in rats, with efficacy studies in rats and canines. We have a few months of molecular cloning, and then about a year of protein purification and analytical process development, and then a couple years in the preclinic testing the formulation on rodents and canines. Here are the project planning Gantt charts indicative of our path to the clinic. It's a reciprocal pharmacokinetic experiment, the pharmaceutics of SODA in the animal labs. It's an important step in advancing the candidate through its selection switches. We're starting a direct to phase 2B mode even in our animal experiments for potential translation to the clinic. A good floor plan, lab layout, and a standard bay lab, which are available at any biotech incubator in the world. We're considering either Sid Martin in Alachua, near Gainesville, the home of the University of Florida, or we can gain strength in our independence and solidarity by moving the project and its military medicine applications overseas to Switzerland. We are also willing to relocate to California or Massachusetts, depending on investor preference. We're running ATCA-alike with TFF and a microfluidizer to make sure product is not just pure and safe, but bioactive and stable as well at fill and finish in the syringe. It's a large sum of our expenses, equipment and materials. The barrier to entry and necessity to run a custom wet lab to arrive at phenomenal product and acceptable IND is a considerable hurdle. We're raising up to $1 million in a seed round and up to $12 million to establish the laboratory in a Series A. I'm Aron Workman, CEO. My contact information is aaron@arowor.com and 754-261-6155.